guys, welcome or welcome back to my channel. Today we are going to discuss another important topic, that is role of p53 gene in as a tumor suppressor gene, right? So basically p53 gene is named so because P stands for protein and 53 is its weight, right? So it is a gene which codes for a protein, that is p53, right? It acts as a tumor suppressor gene because it can cause, why it acts as a tumor suppressor gene? Because it can cause a cell cycle arrest. Its key role is in, first is it helps in regulation of cell cycle. Obviously, if it is a tumor suppressor gene, it can help in regulation of cell cycle. We'll understand its mechanism how, right? After that, its second role is in apoptosis, right? Okay, so after apoptosis, last is we have, it helps in the maintenance of genomic stability. How? By the mechanism of DNA repair, right? So basically it arrests the cell cycle at G1 phase, right? We'll see how, but I'm telling you that it arrests in G1 phase, right? So let's see. We'll study about two conditions. One is normal one, and to contrary to it we have the stressed condition. Stressed condition is the one in which there is mutation. That is how mutation, we'll also learn about that, right? So this is our cell cycle, this is our G0 phase, then we have G1 phase, then we have S phase, G2 phase, and the M phase. Oh, this is the interphase, right? So what happens in a normal condition, that at the in the G1 phase, at the checkpoint between G1 and S phase, the p53 gene codes, is a transcriptional gene, right? Transcriptional gene. So it codes for many proteins other than p53. So it also codes for a protein that is named as MDM2. So in normal condition, MDM2 binds with p53 at the G1S checkpoint. So okay, when it binds with it G1 at G1 phase, when p53 binds with MDM2, so what happens? Consider that this is your p53 gene, and wait, and this is the cap of MDM2, right? When it happens, then as p53 is a protein, so it gets degradation, right? And MDM2 is given sideways, left out, right? So it gets free, okay, right? This is a concept which will later on tell us how it helps in apoptosis, right? Whereas when we consider the stressed condition, right? So basically we'll consider about G1 phase. In G1 phase, when the condition is stressed, p53 get phosphorylated by the it gets phosphorylated because of the mutation caused. And mutation, what is basically the double stranded break created, which cannot be repaired easily, and it is the most common form of the mutation. So it does not bind with MDM2, right? So as MDM2 and P53 binding doesn't occur, so P53 doesn't get degrade, so proteasomal degradation does not occur, right? If it does not occur, it starts checking at the cell cycle, right? If it has started checking, then it arrests the cell cycle at the G1 stage, at G1 stage, right? I hope this concept might be clear till now, right? So now let's understand the major role of P53. This is P53. As here you see that its level increased. How it increased? Because no proteasomal degradation occurred, right? And if no complex formation leads to no proteasomal degradation, and this leads to the increased level of P53. So it might be clear till here that the level of P53 increases. So when the level of P53 increases, it leads to the cell arrest, which prevents the cell entering into S phase is prevented, right? Okay, so basically, so increased level of P53 means that it has been activated. So now let's understand how it basically gets activated. One reason is increased level of P53. Another reason here is damaged DNA, right? Which form of damaged DNA is double-stranded bond breakage. Double-stranded breaks in DNA, right? Okay, so when P53 level is increased because of damaged DNA, it activates its damaged DNA sensor, also known as the maintenance genes, get activated. These are ATM and ATR. They get activated, right, which leads to the activation of P53 by its phosphorylation, right? After phosphorylation, P53 gets activated, and it also leads to the activation of P21. When P53 is activated, it either leads to two cases. Either it leads to the DNA repair, or the DNA remains unrepaired, right? These might be the two cases. Okay, if the DNA remains unrepaired, then it further can be of two types. Either it can lead to apoptosis, or it can lead to the sustained arrest of cell cycle at G1 and G2 phases. It can also get arrested at G2 phase, right? P53 after phosphorylation. Here I shall mark this. Here this acts as a transcriptional activator. Which activates P21, which is also a gene, and when P21 get activated, this is related to WAF gene that is coded by P53 only. This is not that important. If you want to, you can, but majorly you have to learn that P53 activates P21, and this P21 acts on CDK4 and CDK6. Why I have written CDK4 and CDK6 here? Because basically in the G1 phase, CDK4 and 6 are involved, right? So it acts on CDK4 and 6 and hence hinders their function. Function is hindered. So the cell cycle stops, right? Thus preventing the formation of tumor and the formation of cancer. Right? Okay, here I have told you that MDM2 gets separated in normal condition, right? If one more thing here is, if MDM2 is free, P14 ARF can also get bound to it, which is an oncogenic signal. This also leads to increase in P53, or you can say formation of pool of P53 protein, right? Which increases the risk of cancer. Okay, so checkpoints become free. And normal functioning gets disrupted. Normal function gets disrupted, right? You might be thinking that how the checkpoints become free, because checkpoints are regulated by CDK4 and CDK6 where P21 gene acts, right? So it gets become free, leading to the, here the P53 pool becomes increased level of P53 causes cancer. Cancer leads to mutation, and in case of mutation the checkpoints become free and the normal functioning gets disrupted and the cell cycle regulation is also stopped. And that is why tumor suppressor gene is known as the regulator of cell cycle or the guardian of genome. Okay, one point that we forgot to discuss is tumor suppressor gene. Basically what they do, they show recessive mutation, either recessive mutation or loss of function to cause cancer. Otherwise they are tumor suppressor gene, that is they stop the formation of cancer. I hope the topic might be clear to you. If you have any doubt regarding this topic, you may drop down in the comment section. Thank you. Like, share and subscribe.